Hi, my name's Jason McLain from NASA Goddard. Thank you for giving me this opportunity to talk about this work. I'd like to thank Dr. Yo, my NASA postdoc, Sophia Westerkamp, an extremely talented 2023 summer intern in the astrobiology analytical lab at Goddard, whom lab manager Hannah McLain performed. All of those performed the bulk of this work. I'd also like to thank our sponsors, the Survey Node Leader and the NPP program. Spots et al. Discovered amino acids in return lunar samples in the early 70s, and these results have been confirmed more recently. Britton and Beta postulated that approximately 1% of the exogenic meteoritic amino acid and organic composition could survive lunar impact. That's as opposed to softer landings of carbonaceous chondrites on Earth that can conserve even more of the organic content. And Elsila in 2024 discovered small concentrations of amino acids in the ANCSA samples under various degrees of conditions and locations. Several studies have explored the stability of amino acids under lunar conditions. Carl Sagan postulated that the low concentration of amino acids in return samples and organics is in line with impact gardening, and that the half-life of amino acids exposed to lunar surface conditions was about 10,000 years, and that impact gardening would destroy the bulk of these organics, which is in line with the small amount we see in the lunar samples we have. Pascal Freud also studied amino acid ices and shown that they are highly susceptible to UV photodestruction. However, other studies recently have looked at radiolysis of amino acids under various conditions and showed varying degrees of resiliency to degradation, especially for gammas, solar energetic particles, and alphas. However, there's a limited amount of data on the amino acid regolith interaction under UV photolysis. And here we hope to gain some insight whether or not the amino acid molecules can survive UV radiation while bound to the lunar regolith. The focus of this study evolved to determine at what depth does the lunar regolith protect amino acids from photodestruction. The UV photon flux at 1 AU is approximately 10 to the 17 EV per centimeter squared per second. For our study, we used a hydrogen electrolytic hydrogen lamp, which has a flux of about 10 millijoules per centimeter squared per second. If we look at these numbers, that's about a third of the flux that's seen at the moon. And for perspective, the UV fluence for sterilization is on that same order. So we have an experimental setup here to focus on to determine at what depth the lunar regolith will protect amino acids. The experimental setup is quite simple. We use half-inch test tubes coupled to a vacuum system in order to evacuate and irradiate the mixtures at elevated temperatures. The hydrogen plasma can be seen here and is extremely white, indicative of a pure hydrogen plasma that's producing the Lyman alpha UV radiation. There's a blue label on top, which you can see that, but the plasma is extremely white within the microwave cavity, whereas small amounts of atmospheric gases such as nitrogen, the plasma would be pinkish in color. Here's a diagram of our experimental workflow showing the step-by-step process. It's important to note that the process is repeated 3 times for each, the control, the standard, and for the experiment. So that's a total of 9 runs for one experiment. For the first experiments, we simply dropped amino acid standard solutions onto the top of the soil inside the test tube. The soil itself is extracted, and the test tube is also rinsed and extracted. And here, this is why the sample then becomes two separate test tubes. There are several procedures to dry down and concentrate the amino acids before doing a derivation, which here we perform what they call Accutag derivatization, followed by liquid chromatography, mass spectrometry. Here you can see the experimental setup, the Accutag derivatization, UPLC, which is also coupled to the Zevo triple quad. The derivatization has a high infinity for the amino acids and brings the mass out, so you can do very high quality, reproducible, and very sensitive measurements of amino acids. Here in Figure A shows the selected multiple reaction monitoring of the triple quad, and in Figure B shows the fluorescence chromatographs. The first experiments were simply to look at whether the amino acids could be recovered under various degrees of vacuum. So here we performed rough and high vacuum pumping on the test tubes for the controls and the no vacuum samples, as well as rough and high vacuum. And here the results confirm that the lunar volatile, the amino acids are not volatile at lunar temperatures and vacuums. You can see here from the lighter highlighted colors is the difference between the dust extraction and then just the rinse, what is left over in the test tube following the extraction.
2 minutes, Jason.
Yeah. So can amino acids survive this radiation plasma? Well, the answer is yes. But as you can see here, after about 10 minutes, over 80% of the amino acids have been destroyed. But as we irradiate up to 2 hours, you can see that the amino acids persist. This led us to use an isotopically labeled double carbon glycine solid solution to perform a depth analysis in order to determine the survival rate as a function of soil depth. Here's a picture showing the solid solution on the left, and then on the right, we can see where we've dosed the sample right on the surface, and then injected the sample 2 centimeters below the surface. Here we can see that the surface non-irradiated versus the surface plasma irradiated, that we destroyed approximately 60% of the amino acids just after 30 minutes of irradiation, whereas the buried amino acid solid solution was not only fully recovered, it's actually more than the surface not irradiated, which is extremely interesting and leads us to hypothesize that the plasma irradiation potentially changes the binding efficiency of the soil. And therefore, the more irradiation, the more the less efficient the amino acids will be stuck to the soil, and therefore they are extracted more easily. In the conclusions, vacuum conditions have apparently no effect on amino acid stability. The hydrogen plasma radiation does not penetrate the lunar simulant to degrade the amino acids, which has also been seen by Matthewman in 2016, showing very little degradation of amino acids. They survive buried more than 2 centimeters. So future studies will look at a more in-depth depth analysis and explore this amino acid surface binding efficiency as a function of plasma radiation. Thank you.